my cute little first PUC students. I hope every one of you who is watching this video is doing perfectly great, and today I am going to talk about all the diagrams in one particular video. Now this is very important one, but the correct use of it will be analyzed only if you try to draw the diagrams as well. Okay, I mean you have to practice the diagrams really well. So for instance, let me talk about this particular chapter, the first chapter. So I have put according to the chapter weightage, the cell, the unit of life. You have plant cell and animal cell on page number 92 and 92 itself. Okay, now you look at this diagram really carefully, then you close the book, try to draw it by yourself. Okay, now what you have to do is open your book and label it accordingly. Try to label it in the picture that you have made. Now when you label, you will be finding certain structures that you have not made, because you might not be finding where to label that particular structure. Basically, suppose there was something like this in the real picture, but in your picture it's not there. Here in your picture it's blank. So you will be searching for it and probably drawing it and then labeling it. So that is the best way of learning things. So do that, learn from your mistakes and practice these diagrams because these are very important diagrams. So first, as I said, page number 92, plant and animal cell, really important. Go for the labelings. Okay. So whenever you get time, whenever you're getting bored, okay, you can practice diagrams. Now it can be when you were studying physics, chemistry, whatever, you're getting bored of it. Fine, take a 5-minute gap, and what you can do is open this particular PDF and start drawing the diagrams which are mentioned over here. Cool? So next thing, page number 97, there's a diagram of mitochondria, and page number 98, I have put the page number for easily accessing, okay, for you to easily access things. So you can take it from your NCERT as well. Okay, so this is the structure of the chloroplast. You should be knowing about its structure and also its labeling really well. If you're not knowing about the labelings, then you will probably lose a lot of marks out there. Okay, so check for the labelings. So these are the pictures that you need to draw from this particular chapter. Going ahead, plant growth and development. Over here you have sigmoid curve and straight curve which is there, okay, arithmetic growth and logarithmic growth and things like that. So you should be again focusing on this. You would be asked to make the sigmoid curve and show, basically make this graph and show. So have an idea of all these things. Now even if at all they are not asking you directly draw the graph and show, they might ask you about the logistic growth. Okay, explain what is logistic growth. So you obviously have to support your answer with a graph. For that you should be knowing to draw it, right? So please understand that. Please understand the picture really well. What is the concept behind it? Why is there a stationary phase? Because the resources are limited, my dear children. The growth would not be happening. It will be in a limit. It won't be going beyond it, because the resources which are there, it is not infinite. You have finite resources. So over here we assume that there is infinite resources and it's going on. So basic things you should be aware of. Page number 170 and page number 170, again on the same page you would be finding it. Now chemical coordination and integration. Over here you might be asked about the schematic representation of estrogen, a hormone. So this is what you have to draw. This is on page number 249. Please recheck once, okay? Please recheck once in your NCERT. I have used the rationalized version of NCERT for following this, okay? So this is the schematic representation of estrogen, how this particular hormone work. Now this is from chemical coordination and integration. Cool? Next one, locomotion and movement, page number 224. You would be asked to draw the human skull. So you should be knowing what is this. It's a frontal bone, parietal bone, temporal bone, occipital bone, all the things, okay? Lacrimal bone, nasal bone, zygomatic, maxilla, mandible, sphenoid, everything, hyoid bone. What function is performed by them? Where is it located? In that way, or probably if you go to YouTube and all, if generally in any sense, or you just go to ChatGPT and ask them to make a code for remembering the human skull, and it will be giving you a code. In that way you can remember the labelings much more better. Okay, then on page number 221 you have actin and myosin filament. The diagrams of it, again very much important. The question have come, draw actin and myosin filaments. Cool. This is from locomotion and movement chapter. I hope you have gone through the page number as well. Now cell cycle and cell division, you have page number 121, the whole cell cycle. There is G1 phase, S phase and G2 phase. Then there is M phase where the actual division takes place. This whole phase, my dear children, together what do you call it as interphase, and this is where the cell spend 90% of its time. Okay, for example, if I say the cell cycle is of 24 hours, then for almost 23 hours it is in the for hours, not percentage, it is in the interphase. Rest 1 hour it is going to utilize for the actual division up over here. Do not forget to mention G0 phase, that is the resting phase out there. Okay, cool. Chalo. Yeah, this is on page number 121. Going ahead, respiration in plants, very important thing. You have to represent glycolysis properly. So over here, glycolysis, yes. So you have to mention where is ATP being utilized, okay, where is H2O being produced, where is NADH being formed, what is the what are the things, how many carbons are there in each compound which you are making over here. All these things is really important. Remember, from one glucose you are going to get two molecules of pyruvate or pyruvic acid. Okay, so all these things, please make sure you are mentioning it in the schematic representation of glycolysis. Talking about the next thing, that is the citric acid cycle or the Krebs cycle or the TCA cycle. Okay, so over here on page number 159 it is given again. The pyruvate gets inside the mitochondria, you have acetyl coenzyme A, it reacts with oxaloacetate, you get what? Citric acid convert to alpha glutaric acid, then succinic acid, malic acid, and the same thing goes up again. So all this thing along with what is being released, so 2 steps CO2 will be released, my dear children. Okay, in in 2 steps again NADH is being released, in 1 step GTP and 1 step FAD plus is being released. Fine, not in 2 step, 1 to 3 steps NADH is being released. Cool. So all this important piece of information along with the number of carbons which are there in a compound, you should be knowing it. Chalo. And biomolecules chapter, page number 159. Okay, this picture might be given and you might be asked that why is okay, here it is mentioned as A and this might be mentioned as B, and you might be asked why the activation energy is too low, or why is the graph like this. Is it because there is no enzyme or there is enzyme? Does A has enzyme or B has enzyme? All this type of questions can be coming. So you should be knowing the concept behind it. If you are not knowing the concept behind it, you might not know how to draw it or probably how to explain the diagram if it is coming in the form of a question as well. So please go through the concept over here. Cool? So it's not just I am telling you the diagrams which you have to draw, it's also the diagrams which could be giving you concept related questions as well. Now from body fluids in circulation on page number 198 you have a diagram of heart. Now this heart might seem a bit difficult for you to draw. So here we have another heart that I have put up for you. This is not in any page number. I just copied it, which is why it came like that. Okay. So basically this is how it appears. You have right atrium and right ventricle. This is the valve which is present between right atrium and the ventricle, and this thing that you see is another valve that is between the ventricle as well as the pulmonary artery which is there. Here also this is the left atrium and left ventricle. This is the valve you have. Again you have a valve between the ventricle and the aorta, okay, which is this. And obviously no, this is pulmonary vein bringing things up, okay, and the name of the valves: mitral valve, tricuspid valve, and pulmonary valve, aortic valve. Where is aortic valve? Yeah, aortic valve. Yeah, aortic valve. The septum which is there in between it, superior vena cava, the inferior vena cava which is there. All these labelings are really important. Make sure you are drawing it properly. Okay, going ahead. Page number 201, the ECG, echocardiogram. P, what does it stand for? Systole or diastole. QRS, what does it stand for? Ventricular systole, right? Now T, what does it stand for? That means systole is ending. That is what you mean. So this whole thing, you should know the concept behind the echocardiogram. ECG. Oh, yes, where is it? Cool? Chalo, next thing. Breathing and exchange of gases. You have certain important pictures over here. 184 page number, you have this structure of the lungs. Okay, so make sure you know about bronchi, bronchioles, everything, the pleural membrane, the pleural fluid, and epiglottis, larynx, trachea, all the things which are being mentioned up over here. Now this is where the exchange takes place, that is between the alveoli and the blood vessel. So all the things, squamous epithelium is there of the alveolar wall, and endothelium of blood capillary is there. There is basement substance between that. Okay, all this red blood cells are there. Okay, so this thing is important. Again, you have an oxygen dissociation curve. You might be asked to draw this. Okay, this is on page number 189. Next, photosynthesis in higher plants, my dear children. So this is what? This is your C3 cycle, Calvin cycle. So you might be asked to make the Calvin cycle. So when you're asked like that, you have to draw it. So this is the Calvin cycle, or you call it as a C3 cycle, the dark reaction, whatever. And now this is what? The C4 cycle. So you might be asked to draw that also. In C4 cycle, there are two compartments where things are happening. Here it is mesophyll and this is bundle sheet. Okay, so the actual cycle event of things happens over here. Here fixation of carbon dioxide happens in some other form. They convert into what's a malate and things. Okay, so please be aware of all these things. 144, 146. Now talking about same chapter, 139, you have non-cyclic photophosphorization. Over here you have cyclic photophosphorization. So make sure you know about this. Okay, this is a Z scheme basically. Okay, that is a light reaction, and this happens in bacteria and all the other things. Okay, 139 and 140 page number. Look for the terms which are given up over here. Photosystem 1, photosystem 2, light is coming, light harvesting, it is falling on light harvesting complex. Electrons are going to go into the high energy levels. They are taken by the electron acceptors, and ATP is being formed, NADPH is being formed, and this NADPH, ATP which is being formed, it is used in C3 cycle for the formation of what? Glucose, my dear children. So all the things are really important here also. The same thing happens in a different way obviously. So you should be knowing about this as well. Now, excretory products and their elimination, you have page number 207, the structure of a nephron, a very important picture. So no efferent arteriole is coming, efferent arteriole will be coming outside. Okay, this will be broader, that one will be kind of narrower because blood comes in, what's say, high pressure, no? So this would be taking lots of blood, but over here filtration has to happen. So what happens? This is being narrowed down. The efferent arteriole will be narrowed down. Why? Because you want the blood to stay here for some time. If it's narrow, not all the blood will be going around very fast. If it's narrow, the blood will be staying here for some time, and then slowly it will be moving. Okay, so that is the thing. So glomerulus, Bowman's capsule, PCT, DCT, descending limb of Henle, and ascending limb of loop of Henle, all this thing you should be knowing. Descending, it will be a bit kind of thicker over here and then thinner. Here it will be thinner, it will be thicker that side, and the collecting duct as well. Cool. Chalo. Next, morphology of flowering plants, my dear children, the structure of the root tip. Okay, the region, different regions. So region of maturation, the root hair, region of elongation, region of meristematic activity, and the root cap and everything. Over here you have aestivation, page number 64. Again, you might be asked about aestivation. What are the type of aestivation? Just like how you are asked about type of placentation, right? So you should be knowing the diagram. If you know the diagram, you might be able to write it better. Cool. So for page number 59 and page number 64. Now here you have the structure of the monocot embryo. Okay, this is what the structure of monocot embryo, my dear children. Okay, page number 59. So you have scutellum, coleoptile plumule, radicle and coleoris which is found in them. Okay, and endosperm. Monocots are the one where you will be finding endosperm as well as a cotyledon. In dicots, the endosperm is totally utilized for the formation of cotyledon. Now this is the floral diagram of Solanaceae family. Solanaceae family. So please know about that. Then you have the structure of leaf. So please draw axillary bud, leaf base, petiole, stipule, lamina. Okay, cool. Page number 64. Okay, cool. 59, 68, 64. Going ahead. Neural control and coordination. This is the structure of a neuron. So again, dendrites, the Nissel's granules, okay. So in them, they have a modified RER, that is rough endoplasmic reticulum in the case of neurons, because they have to make certain very important proteins. So that modified RER is Nissel's granule. You have cell body. In the cell body, you have the nucleus. Then there is a Schwann cell over here, my dear children. There is axon, and there is myelin sheath. There is nodes of Ranvier. The point where the myelin sheath is absent, you call it nodes of Ranvier, okay. And then there are axon terminals and also a synaptic knob. And over here synapses. At the synapses, what is happening? You should be knowing about that. This is on page number 234. The neurotransmitters are being released. So synaptic vesicles comes over here, fuses with it, and releases the neurotransmitters in where? In the synaptic cleft. And then in the postsynaptic membrane, these neurotransmitters are going to attach on the receptors, my dear children. So you might be asked to draw it and also explain it. So please know how does the flow exactly work. Okay, 234 page number, that is about synapse. Now, anatomy of flowering plants, you might be asked to draw the diagram of stomatal apparatus. This is the thing. Okay, the subsidiary cells are right over here, and then you have the guard cells, then you have the stomatal pores. In the guard cells, obviously you have the chloroplast, okay, and then there are certain epidermal cells also outside the subsidiary cells. So please know about them. That's all. Yeah. So that's the open and the closed stomata which is being mentioned over here. Now in biology classification, the structure of nostoc is important. It is there on page number 13. Okay, heterocyst and mucilaginous sheath which covers the whole body of it. Now structural organization in animals. Okay, I have messed up with this particular thing. That is on what page number does it fall? Can you just give me a second? I'll tell you what page number does it fall. Okay, just give me a second. Over here I have it. Yes, so it's on page number 81. So I don't want you to be stranded because of this. So this is the complete digestive system of the frog, my dear children, and it's on page 81. You can just go through it. This is from the chapter Structural Organization in Animals. So will you be able to do all these diagrams? I know that you would be able to draw. Just as I said, what is the first thing that we need to do? The first thing is analyze the picture really carefully. Okay, look for the labelings, know the concepts, be sure of the concept. When I say this, analyze the picture, I'm trying to say be sure of the concept behind the pictures. Then you would be able to relate and learn. Then what do you have to do? Close your book. You have to close your book, draw it, and label using the book. Okay, now when you label using the book, you might find that you have not drawn certain things, so you will draw it. Now when you draw it again, probably this time it will be perfect. So this time you are not going to label it using the book, you are going to try to label it by yourself. Label it by yourselves. So in that way you are gonna ace it, my dear children. So only 2 diagrams are important. Okay, 2 times you have to draw it, but in these 2 times if you draw it properly, as I said, in the manner that I am saying, it will be really useful for you guys, which is why do not miss anything. Okay, and also the bacteriophage diagram is important. Okay, just go through it once. Tobacco mosaic virus and bacteriophage diagram, draw that as well. Cool. And with that, we complete all the important diagrams of first PUC. If you need my help in anything, do let me know in the comment section. I'm wishing you all the best. Your exams are going to go really great. Bye-bye, my dear children.